Good evening, Professor. This is my personal timetable. So in the next week, I have planned the sixth task. The first task is PSA biosensor paper writing that is already submitted in Advanced Sciences. And the task 2 is urine biosensor research. So to find the solution for the better attachment on the surface, and we also did AFM, SEM in the last week to check the surface and what we got in the AFM and SEM. I will explain you later. And the task 3 is the review paper. Is already modified some content and need to be updated the figure. It's edited by the Jaya, and I will submit to you on this Friday. The task 4 is corona biosensor research. Me and Prashant will discuss about the how to immobilize antibody and antigen and what and which concentration, and also we start today the research of the coronavirus, and hopefully we will submit to you the some results in the next week live meeting. And the task 5 is AAO biosensor research. I have discussed with the Bruce about the new research plan because uh, he did some LSPR and SPR measurement, and I thought it was wrong concentration. So we have discussed and make a new research plan. And the task 6 is the meeting with Jacob and Bruce about his research plan, and also make a draft paper writing. So this is the research plan of urine PSA biosensor, and it's already almost done. The 70% experiments is done, and the 30% is remaining in the terms of water contact angle measurement. And uh, on this on the next weekend we will go to the cans for contact angle measurement. We have three type of antibody: 1H125A6, AB2218. We have three type of antibodies, and one is antigen. It's a native human prostate specific antigen protein. So I have to just separated out the three antigen that what we have antibody in our lab. So I just want to classify and to finalize at what type of antibody is best conjugated with the specific antigen that what we have in our the refrigerator. So with the help of capacitance, we define that 5A6 is not appropriate for our research, so I just exclude this antibody, and apart the rest of the antibody, that is 1H12 and AB2218, we move to the forward step. And then we had a FTIR of the three antibody. Here was also signified that the 5A6 is not suitable for our research, or is not 100% binding with the antigen. So we go with the two antibody, that is 1H12 and AB2218. So this is the FTIR spectrometer. We have checked the FTIR spectrum in four different kind of samples. First one is the bare chip. It's just after a piranha cleaning that we got the three type of peaks. It's already shown in the figure. It's a different kind of signage. And the second one is treated with EDC. So we got six type of peaks, and it's already mentioned here. It's just carbon-carbon bond, OH bond, NH stretching bond is that is called secondary amine bond is present in the after the immobilization of EDC NHS on the surface. And then is the third one is a 1H12 antibody. This graph signifies that we have the NH stretching secondary amine bond, amine salt, conjugated alkene, and nitro compound, and phenol as present in our sample. That is the presence of a 1H12 antibody. And we have the same kind of peaks in the AB2218. It defines that the antibody act the same on the surface, because the antibody combined with the antigen, and then we got the this kind of different peaks. So here is also combine the three different kind of samples in a one frame, and then we check the different kind of peaks, they have any changes or not, in a step by step mode. So the bare chip is the black line, and the red one is EDC, and 1H12 is the blue line, is functionalized with the antibody and antigen. So we got NH12, NH2 and 3328.95 the same peak, and NH bond also present in this peak, and CHC double bond C stretching on the 164.47 peak, and 1556.89 which shows the NO bond, and the rest the OH hydroxylic bond is 13676.75 which shows the transmittance changes in the terms of wave numbers. So here is also the different kind of antibody we used, and to conjugate it with the antigen and to show the FTIR peaks. So this is also the same kind of result that what we got in the case of 1H12, but the peak difference, the transmittance percentage is different. So it signifies that our antibody is perfectly combined with the antigen, and to get this kind of peaks to some easily combined with the EDC and on the surface, and the bare act like a black line. So this is the AFM microscopic morphological images. The first one is a bare surface, is taking the image is at a 10 nanometer, and the second one is the bare surface with graphene oxide, the same scale 10 nanometer, and the third one is bare surface with GRO and having a 1H12 antibody in the presence of EDC NHS. So we got the 1H12 antibody, shows some red color patches on the surface and the circle kind of star like shining. So we got so we signify that the antibody is easily combined with the antigen. So and also the other side is AB2218. It shows the presence of antibody easily combined with the antigen in the presence of EDC and graphene oxide. So this is the morphological images of with the antigen. So it shows the kind of branches on the surface. It signifies that the presence of antigen, and the red patches and the dotted kind of surface on the chip, then we with the help of antibody. And the other side is AB2218 antibody, the different kind of antibody on the same antigen, is a 100 nanometer scale. Then we also got the same kind of peaks in the surface morphological things on the chip, and the red patches in the circle cluster on the surface. So we got the cluster on the surface, and it shows the perfect binding with the antibody. So sir, this is the SEM images. It's a bare image, bare surface image, and this one is the bare surface with graphene oxide. Graphene flakes shows on the surface, and this bare surface with graphene oxide and with EDC. EDC is basically act like a cross linker. So just fix or to bombard on the surface to cross link between the bare graphene oxide and the EDC, and it's also the some lone pair kind of things that easily combine with the antibody and antigen. That's why it shows the hollow structure, and you have seen the branches kind of in a 200 micrometer for high quality images. And the bare surface, graphene oxide, EDC and 1H12. So this is the 1H12 antibody. In this presence of this antibody, we shows the branches, structure. As you also seen in the AFM images also, we got the branches kind of shining on the surface, so we signify that our antibody is on the surface. And the simultaneously also same in the AB2218, we got the same kind of structure branches, and it shows the antibody is on the surface and it act like the same. So this is the image of 1H12 with antigen. Here the antigen is perfectly bind with the antibody, and you see this square shape on the surface. This is the antigen, and the branches, the 4 branches, is the antibody. And also the same thing is the AB2218 with antigen. It's also the same thing that what we got, the square rectangular shape on the surface is the antigen and molecule, and the branches is act like a antibody molecule. These images can be done in the 50 micrometer, and the 1H12 antigen images is 200 micrometer. So this is the cross section of the sample characterization, that what we did before. So we just cut it out from the middle and to check the cross sectional view, and so we got the length of the layer, that what we have, the platinum and titanium layer. So the platinum and the titanium is almost 200 or 0.2 micrometer, and then with the presence of 1H12 we got this kind of morphology in the cross section part. And the same thing is with the antigen, 1H12 with antigen. So this is the platinum and titanium length, and then this collapse and the seizure kind of stimuli enabled kind of things is on the surface. And this is the image of AB2218. So this is the length of the platinum, and the titanium is almost 0.2 micrometer, and this is the mountain kind of morphology, that is the presence of AB2218 and antigen, they combined together and to make a more thicker surface. Sir, and this is the research plan of my coronavirus research. We will have to use the P3, P6 and D3 chips, and then we follow the following steps. We just change the antibody and antigen here, otherwise this method is almost same as the urine biosensor. This is the antibody images and data sheet, and the instrument we use for characterization is capacitance measurement, AFM, SEM, water contact angle measurement. Then this is my paper writing plan. It's a urine PSA biosensor. So I just use like research highlights, advantages, what are the objectives and methodologies, and also I'm also written some part of the urine biosensor in the draft file. So thank you, sir.